attaches the organs to the surrounding area. So you see a picture like this, excuse me, and you will also see a slide in lab where you've got the mucosa, the submucosa, the muscularis, and then the serosa. So you've got these layers in all the organs of the digestive system. Now, one layer may be more significant than another, depending on what organ you're talking about. And here's a slide, and your slide in the lab is gonna look very much like this. So mucosa, submucosa, the muscularis, and then the serosa layer is on the outside. All right, what controls digestive system? There's nervous control, and we have reflexes. You have a short reflex and a long reflex, and basically they are what they say they are. A short reflex can cause a change very locally. A long reflex can come from part of the brain and initiate a reflex within the digestive system. And again, the parasympathetic is going to dominate. Hormones are going to be key in the function of the digestive system. It's a hormone factory, and we will go through many of those in the next section when I lecture in class. Types of digestion: there's mechanical digestion, which is the physical breakdown of food into smaller parts. You're not changing the chemistry. The most easily understood one is chewing. You're taking a big cookie and making it into small parts of a cookie. Another example is bile. Bile coming from your liver will emulsify fat, make big blobs of fat into small blobs of fat. What mechanical digestion does is it sets up in a situation where chemical digestion will work better, where we're going to take enzymes and chemically break down the food. So mechanical digestion increases the surface area in which these enzymes can work, so they work better. So enzymes will take the big molecules, make it into small molecules, so you can absorb it into your bloodstream. Enzymes, what do you need to know? They're proteins. They speed up chemical reactions. An example is if you ate a steak dinner with a steak and a potato and salad, you could digest it, but it would take about a week without enzymes. With enzymes, you can digest it within 24 hours. They have a very distinct shape to them, and over here I show you sucrase, which is an enzyme that breaks down sucrose. Notice this enzyme fits very specifically in here to break this molecule down. If you change the shape of an enzyme, then you change its function. And so each enzyme has an optimal temperature and an optimal pH at which the enzyme will function. If you change the temperature or you change the pH, then you will change the shape of the enzyme and change its function. It will not work. This is why high fevers can be so dangerous, because they can change the shape of enzymes in all proteins in your body, like hemoglobin and neurotransmitters in your brain, and they will not function. If you change the shape, you denature the enzyme, which means you're going to change the shape. That's what the word denature means. So here we have sucrase. Comes in and it breaks down sucrose into its monosaccharides of fructose and glucose. These two can be absorbed, go through the bloodstream, and be used as an energy source or stored. Enzymes are going to end in ASE or IN. Ones that break down proteins tend to end in IN. So if you see a big long word, blah, 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 ACE, if you know nothing, you know that it is an enzyme. And this just shows you another picture of an example. Here's your substrate. It's just talking about the molecule, so this would be sucrose. Here's the enzyme working to break it into the products of fructose and glucose. So make sure you review these two podcasts on nutrition. Again, I will not cover them in class, but you can come to me with any questions. You can ask me in class because other students may have the same questions, so make sure you understand it. I've also given you a handout, a worksheet, to make sure that you go through this process, because you will be tested on this material.